A restriction enzyme, restriction endonuclease. Definition: restriction enzymes, also called a restriction endonuclease, is a protein produced by bacteria that cleaves DNA at specific sites along the molecule. Restriction endonuclease cut the DNA double helix in very precise ways. It cleaves DNA into fragments at or near specific recognition sites within the molecule known as restriction sites. A restriction endonuclease cut the DNA double helix in very precise ways. It cleaves DNA into fragments at or near specific recognition sites within the molecule known as restriction sites. They have the capacity to recognize specific base sequences on DNA and then to cut each strand at a given place. Hence, they are also called as molecular scissors. Eco-RI restriction enzyme is there. And source of restriction enzymes. The natural source of restriction endonucleases are bacterial cells. These enzymes are called restriction enzymes because they restrict infection of bacteria by certain viruses, bacteriophages, by degrading the viral DNA without affecting the bacterial DNA. Thus, their function in the bacterial cell is to destroy foreign DNA that might enter the cell. The restriction enzyme recognizes the foreign DNA and cuts it at several sites along the molecule. Each bacterium has its own unique restriction enzymes, and each enzyme recognizes only one type of sequence. Recognition sites are there. The DNA sequence recognized by restriction enzymes are called palindromes. Palindromes are the base sequence that read the same on the two strands but in opposite directions. For example, the sequence on one strand is GAA-TTC, read in 5' to 3' direction. The sequence on the opposite strand is reverse, and read in the 3' to 5' direction. But when both strands read in the 5' to 3' direction, the sequence is the same. The palindrome appears accordingly like this, shown like this. Then in addition, there is a point of symmetry within the palindrome. In the example, this point is the center between the 80. The value of restriction enzyme is that they make cuts in the DNA molecule around its point of symmetry. Some enzymes cut straight across the molecule at a symmetrical axis, producing blunt ends. Of more value, however, are the restriction enzymes that cut between the same two bases away from the point of symmetry on two strands, thus producing a staggering break. The mechanism of cleavage of restriction enzymes. When a restriction endonuclease recognizes a particular sequence, it snips through the DNA molecule by catalyzing the hydrolysis, splitting of a chemical bond by addition of water molecule of the bond between adjacent nucleotides. To cut DNA, all restriction enzymes makes two incisions, once through each sugar phosphate backbone, each strand of the DNA double helix. Then types of restriction enzymes. Traditionally, 4 types of restriction enzymes are recognized, designated 1, 2, 3, and 4. Each differ primarily in structure, cleavage site specificity, and cofactors. Type 1 enzymes cleave at site remote from a recognition site, require both ATP and S-adenosyl L-methionine to function, multifunctional protein with both restriction and methylase activities. Type 2 enzyme cleave within or at least specific distances from recognition site. Most require magnesium, single function restriction enzyme dependent on methylase. Type 3 enzymes cleave at sites a short distance from a recognition site, require ATP but do not hydrolyze it. S-adenosyl L-methionine stimulates the reaction but is not required. It exists as part of complex with a modification methylase. Type 4 enzyme target modified DNA methylated and hydroxymethylated and guanosyl hydroxymethylated DNA. Now let's learn about nomenclature of restriction enzymes. Since their discovery in the 1970s, many restriction enzymes have been identified while type 2 restriction enzymes have been characterized. Each enzyme is named after the bacterium from which it was isolated, using a naming system based on bacterial genus, species, and strain. For example, the main name of the eco-RI restriction enzyme was derived as Escherichia genus Co. E for Escherichia genus, Co for coli, specific species. Then RY13 strain. I first identified one. Order of identification in the bacterium RY13 and 1 strain and first identified order of identification in the bacterium. Applications of restriction enzymes. Restriction enzymes can be isolated from bacterial cells and used in the laboratory to manipulate fragments of DNA such as those that contain genes. For this reason, they are indispensable tools of recombinant DNA technology or genetic engineering. The most useful aspect of restriction enzyme is that each enzyme recognizes the same unique base sequence regardless of the source of DNA. The most restriction enzymes can be isolated from bacterial cells and used in the laboratory to manipulate fragments of DNA, such as those that contain genes. For this reason, they are indispensable tools of recombinant DNA technology or genetic engineering. The most useful aspect of restriction enzyme is that each enzyme recognizes the same unique base sequence regardless of the source of DNA. It means that these enzymes established fixed landmarks along an otherwise very regular DNA molecule. The most useful aspect of restriction enzyme is that each enzyme recognizes the same unique base sequence regardless of the source of the DNA. It means that these enzymes established fixed landmark along an otherwise very regular DNA molecule. This allows dividing a long DNA molecule into fragments that can be separated from each other by a size with the technique of gel electrophoresis. Each fragment thus generated are also available for further analysis, including the sequencing. One value of cutting DNA molecule up to discrete fragments is being able to locate a particular gene and fragment where it resides, which is done by the general technique of Southern blotting, performing Southern blotting.